Hello. Should I select the incorrect or the correct one? I think option B, biosafety hood, is the incorrect. Uh, so according to me, it's like, biosafety hood is specifically uh, when we enter the laminar or um, when we are handling microbes that have risks for causing diseases. So there are different levels of biosafety levels. So I think it is related to the uh, type of biosafety that we mostly use when we enter the laminar. So this biosafety hood is not basically related to the strong acid or other fuming chemicals. So I think it is not correct.
Okay. Okay. Is there some? Okay. You also mentioned about laminar and biosafety interchangeably. So, do you think if there is any difference between biosafety hood and laminar hood?
Yeah, I think it's quite similar, but I can't exactly differentiate right now. But
Okay. Okay. Good. How do we pronounce your name? Sorry, I can't pronounce your name. I don't know the right.
my name is Sanjanam Singh.
Sanjanbam. Okay, that's easy to pronounce. Okay. Good. Anyone else? Do you want to talk about another incorrect option? So Sanjanbam had actually mentioned two incorrect options, both laminar hood and biosafety hood. We'll come to the difference in the next slide. But there is at least one more incorrect option. Does anyone want to try? Yes, Sanjanbam. Do you want to answer or? Okay, I'll go to the. So the other incorrect option is exhaust fan. So the right answer is actually chemical fume hood. We'll take a short look at what these different terminologies mean. So first, a laminar hood is something that looks like this. So what you have here is from within the hood, you have air that is being blown towards the worker to admit the other person. So if you see in a laminar hood, you have air coming from inside that is blown towards the person who is using the hood. This ensures that nothing enters the cabinet. So whatever sample is inside is not contaminated. Whereas in a biosafety cabinet, you don't have air blowing towards the user. In the biosafety cabinet, the worker or the person who is going to use the hood is prioritized. So to protect the worker, the air from inside is passed. It is actually sucked in. This is similar to how chemical hoods also work. In a chemical hood, you have a similar setup. Because there is a negative pressure, whatever fume that you have within the chemical hood is sucked in. So this way, no chemical that is inside the hood will reach the user. This is just a small video that shows how it works. So the moment you close the lid, you will be able to see that most of the fume enters inside because there is a negative pressure. So, so it is drawn towards this region. So that primarily is the difference between these three hoods. Moving on to the next question. Biosafety level equipment. Which biosafety level equipment is required to work with Ebola virus? 4, 3, 2, 1. You can think about this question for a couple of minutes and then uh, raise your hands. We can discuss what are the 3, 4 different biosafety levels. Uh, Please do not type the right answer in the chat box. It would be better if we could discuss the options so that everyone can learn. And we can start by discussing the incorrect options. You can take a couple of minutes to think about the question, then raise your hand. So far we have two people. Each of them will talk about one particular incorrect option. We'll start with Divya Darshini. You can talk about one incorrect option. Yeah, Divya Darshini, you can unmute and talk about one incorrect option.
Hello, sir. Am I audible?
Yeah, we can hear you. You can talk about one
Yes,
incorrect option.
okay, sir. Sir, biocabinet level is porous and for Ebola virus because it's
So you can talk
highly.
about an incorrect option. Do you know
Oh,
about
okay,
the other
incorrect.
options? Yeah.
Okay.
Hmm.
Okay. Can I talk about three?
Yeah, you can.
Okay. Fine. Uh,
You can tell us for what kind of organisms BSL level 3 is needed.
maybe it's used for like respiratory microorganism work in cabinet 3 like salmonella typhi like airborne disease. We can use it for airborne disease, salmonella typhi, like that kind of microorganisms.
Okay, okay. Okay.
Mm.
What I know about BSL-3 hoods is that in BSL-3 hoods you can work with uh, SARS viruses. You can
Yes,
also
yes,
work with medically relevant Staphylococcus aureus strains. You can work with medically relevant Mycobacterium tuberculosis strains.
and
These.
Musco,
Sorry?
Musco.
Okay, Moscow. Okay.
Yeah.
Hmm. Okay. Good. Okay.
Thank you.
Thank you. Does anyone else want to talk about other incorrect options?
Can I talk about BSL one?
Yeah, sure.
Yeah, the like this is for non-pathogenic, like not highly risked microorganism
Okay.
like E. coli, Bacillus subtilis, and Saccharomyces cerevisiae, like that. We can use for regular sticking and culturing for basic microorganisms,
Right.
like which is not most
Generally regarded as
pathogenic.
safe. Yeah. These have a certification that these are generally regarded as safe, and you do not need much of an expertise to handle this. And these do not cause skin infections or are not
Yes.
airborne. Yeah. These are the organisms that work with uh, that you can work with in PSL1 strains. Right.
Okay. Thank you.
Yeah. Does anyone want to talk about BSL2 strains?
Again, Divyadashini, can I talk
Yeah,
about
sure,
it?
sure. Yeah. And do you know if there is any differences in the level of protection that people wear?
Yeah, we have to like for basic we just need like normal gloves and like aprons and this kind of thing for basic levels. When we go into skin irritants and respiratory irritants, like that pathogens, like most dangerous pathogen, we have to use PPEs. We have safety. Ethically, we have to use PP for anything. That's why we can't get any infections. Basically, I'm a microbiologist. That's why.
Okay, right. Like you mentioned, for BSL1, all you need is a pair of gloves.
Yes.
For BSL2, you need a closed space that is not
Yes.
open and that is specifically.
For HIV, like that viruses, we use BSL2 for HIV virus TB.
Okay,
Yeah.
okay, okay. And in BSL3, again, what if you don't just require gloves, you also have the setup where your hands and arms are covered.
Yes. Yes. Yes.
Whereas in this is how BSL3 looks like, where you have a way to position your arms, whereas in BSL4 your entire body is covered with a PPE. That's what these people are wearing. So it depends on the sensitivity of the strain that you're working with. So these are the four different differences that you have. And Ebola virus being highly pathogenic requires BSL4.
Yes, sir. Thank you.
The next question is disinfection is carried out with, and four options are given here: soap, hydrogen peroxide, quaternary ammonium salts, water. You can think about this question for a couple of minutes and then start by thinking about the wrong options. We'll eliminate the wrong options so that we get to discuss about the wrong options, and then finally we'll come to the right option. So whoever knows the answer can think about the question for a couple of minutes and then raise your hands. Please do not type the answer in the chat box. Yeah, we'll go with Shreya first. You can talk about one incorrect option.
So the wrong answer here is soap. Soap
Okay.
is basically a detergent which is used to clean the surface. It is used for sanitization purpose.
Okay. So what exactly is disinfection?
So disinfection mainly deals with surface cleaning and inactivation of microbes.
Okay. When you say inactivation, do you mean it kills the microbe or
No, no, sir. It makes it harmless.
by? Like, why would, how would something make it harmless? Is it alive or dead?
So I think it's alive.
Alive?
Sterilization is something in which they are completely non-viable.
Okay. Okay. One thing that you mentioned was right. Here, disinfection is usually specifically meant for some particular surface, whereas sterilization is commonly used, and in sterilization, you want it completely free of microbes. Disinfection is just for you to ensure that there is nothing pathogenic that is there on the surface that you are working with. It could also uh, an example that would come to your mind is when you apply ethanol on surfaces to wipe surfaces just to ensure that it is clean. That is disinfection. And okay, okay. So you're saying soap is an incorrect answer. Okay. Divya Darshini?
Sir, yes, sir.
Yeah, you
Sir,
can talk about one
actually,
incorrect
it's
option.
all the above. Because we use soap regularly to disinfect our hands, but like bacteria won't, like I can't say it won't alive, but it's present, but their spores will be present in our body naturally, because we are roaming around out and in, so we have spores come to, yeah, we, of course we have spores in our body for from the bacteria, fungi, as well as from the microorganisms. So we can use soap for a bath and like as well
Does
as
soap
washing
kill
our hands.
microorganisms or does it remove microorganisms?
Sir?
Does soap remove microorganisms or does it
No,
kill
no, no. It
microorganisms?
won't remove microorganisms, but it can be remove the spores that outside the surface. So we can at least.
No, but does it kill the microorganism?
I think like no, maybe.
Okay, so the thing is with soap, what happens is all of these are hydrophobic. You have these detergents that are hydrophobic with this long fatty acid tail that forms your uh, that are basically your esters. So these would in a way interact with your hydrophobic cell membrane and then just pile it up and just remove your bacteria from your hands like you mentioned. Water doesn't. What about hydrogen peroxide and quaternary ammonium salts?
So hydrogen peroxide also remove the microorganisms
What is
because
hydrogen peroxide?
sir, we are using in like oxidase test to see like the reaction of the bacteria. So it's in also like in first aid kit we always used to have hydrogen peroxide.
Okay.
When we getting wound, we will use hydrogen peroxide first to remove or kill the bacteria.
Is it to suppress the bacteria or to kill the bacteria?
It kills bacteria.
It kills bacteria. So it is bactericidal, not
Yes.
bacteriostatic. Right. So hydrogen peroxide, it's an oxidizing agent. So what it does is it creates reactive oxygen species within the bacteria, which is why you use it for use it as a marker for oxidative stress. So it
Yes.
kills the bacteria. What about quaternary ammonium salts?
Maybe I don't know about quaternary ammonium salts. Maybe I can.
Okay, chemically you can think of it as ammonium. Instead of NH4 plus, you have this N plus, and all the four hydrogens are replaced by some substituent. Let's say a methyl group, something of that sort. So all the valency of this nitrogen is being satisfied by some chemical moiety which is not hydrogen. That is a quaternary ammonium salt. So
Okay.
specifically, you might have heard of these in terms of CTAB, which is used to lyse plant cells. When people want to extract genomic DNA from plants or cDNA or want to extract mRNA from plants, they usually add liquid nitrogen to plant cells or a particular tissue from plant. Then it's frozen. Then they crush it and add CTAB. So this is the quaternary ammonium, one of the quaternary ammonium salt that is used. So according to the and water, it doesn't help with disinfection. So we just need to come to a consensus as to what disinfection is. So if anyone would like to talk about how disinfection is different from sterilization, that would also be helpful. You can just try talking. That's what everyone is doing. You need not be very well qualified to know what a particular phenomenon is. You can attempt and we can discuss. That will be helpful for everyone. Okay, so Nishat, I was asking. So we were discussing about what different mechanisms or different terms would mean. One of these term is sterilization. The other is what we have in context here, disinfection. So if we can decide what disinfection is, then the answer would be easier to know. We have understood that hydrogen peroxide could cause reactive oxygen stress, and like the way Darshini mentioned, it's present in even first aid kits, and it's uh, brought when you have wounds or cuts, which I'm not aware of. I just learned now. Whereas quaternary ammonium salts, these are helpful in lysing cells, and I've known that we can lyse plant cells with CTAB, which is one common uh, chemical that people use for plant cell lysis. So now the question is, how would you define disinfection? Does it have to do with killing the cells or just ensuring cells are removed from a surface? We just need to come to a consensus to that. So, Ruhu Nishat, yeah, you can maybe discuss on that. Ruhi Nishat. Right. So you're saying disinfection is cells are removed from a surface. That's what even I would assume. So yeah, so for that purpose alone, I would say soap would be the right answer. Although in the prescribed text and the questionnaire, they say quaternary ammonium salt. I would say soap because soap doesn't kill, but rather it just removes. Similar to how you have for other detergents and strong detergents would kill. So when you talk about soap, the concentration of soap matters. Yeah, Shreya, you can unmute.
So then how will we differentiate between sanitization and disinfection?
Okay, in sanitization, I would think it is for you to kill, not remove. Or I might be wrong. I don't know the exact difference between the terminologies. I can check up and I can tell you. But I
Okay.
know that sterilization involves killing, like completely eliminating all the bacteria, including spores. Right. What I have given here is this, that disinfection means eliminating the microorganisms from surface. So here, this is what the questionnaire says, that quaternary ammonium salts could be used as disinfectants. And here I've just mentioned what I know about CTAB, which is one of the cationic surfactant, which kills cells. So the next question is the F in the given equation, V is equal to E naught plus 2.303 RTF. Here, what does F stand for? That's the question. So I'm not sure if you would be knowing about the different options that are shown here, but rather you can think about this question in terms of what this equation is. So anyone who knows the name of this equation or uh, what this equation has been named after, or which person it has been named after, you can mention that, and what is the relevance of this equation. You can mention that. So for this question, you can start with mentioning how is this equation referred to, on whose name it's been named after, and what's the relevance of this equation. We'll take it from there. Yeah, Shreya.
Sir, I think this is Nernst equation.
Can you repeat? Sorry?
Nernst equation.
Yeah, Nernst equation. Okay, what is the relevance of that?
I think this is used to calculate the electrode potential. Basically, in pH meters, we can use, I think.
Okay. Do you know something more about this?
No, not much.
Okay, good. Uh, Others? Nishita, Ruhi, Nishat, you can unmute. So what we have, yeah, sorry, you can unmute, and then I'll continue.
Hello, sir.
Yeah.
So this is, I think this is Faraday constant, sir. So V, that's a voltage, I think so, and E is the relationship between voltage that and the electric charge, and the at a particular pH, that means it's talking about some molecule, or the velocity of molecule, or the speed of molecule by which it is traveling. Maybe the relationship between the
I didn't get you. Where did you get velocity or speed from?
not velocity, so it's V, that is a voltage.
Okay.
That then the E, that is what do you say, based on the electric charge. E, that is that stands for electric charge, or the movement of electron maybe.
Okay.
And here I think pH that signifies that amount of maybe hydrogen, positive hydrogen ion which is present, or it is talking about some molecule.
Right.
um, and RT at a, I think T, that stands for particular temperature.
Right.
So altogether we can say, sir, this uh, seeing that the relationship between voltage and current, and the molecule, the relationship, I guess this is a Faraday constant, sir.
Yeah, that is Faraday constant. That is, I would agree to that.
Okay, sir.
Right.
This much I understood, sir,
Okay.
with this
Okay.
with this equation.
Hmm.
Okay, sir.
Okay. I will go back to something familiar to you. So here, if you would see, this is something that you might have seen as Nernst equation, where this E is your reduction potential, this E naught is your standard potential. Your R is your gas constant, T is your temperature, N is your ion charge, and F is your Faraday constant. And usually you have ln Q, where Q is your reaction quotient. Now it helps to calculate how much reduction potential a particular reaction has. So usually you would have seen this in cells, half cells or full cells, to measure the standard electrode potential at a given temperature and condition. And that has been modified in for you to get, to relate it to pH here. That is why you have the term pH. So when you consider ln Q as ln of the concentration of H, which you can rewrite it this way, we all know that pH is pH tells you about the concentration of hydrogen ions, and ln of concentration of hydrogen ions can be given this way, which is why this equation is written and modified this way, the one on the top. Right. The next question is, sodium error gives option A falsely higher pH, option B no change in pH, option C falsely lower pH, option D minor deviations in pH. So before we go to this, we need to understand what sodium error is. So if someone can define what, or can discuss what sodium error is, then we'll come to the different options that are given here. Yeah, does anyone want to talk about what sodium error is? You can raise your hand. Yeah, Ashvima. You can mention what sodium error is.
So basically, the glass electrode in the pH meter, it is designed to take up the hydrogen ion concentration. And when there is sodium concentration more than hydrogen ions, it falsely takes up the sodium ion concentration and gives the readings. So that is sodium error.
So, when would this electrode take up, consider the sodium ion instead of hydrogen ion? Is it something? Does it have something to do with the concentration of hydrogen ion or hydronium ion?
Yes, when the hydrogen ion concentration is very less
Okay.
in the solution compared to the sodium ions, then it does.
Okay. And why do you think it happens? Does it have anything to do with the way sodium ion is or hydrogen ion is?
I'm not sure about it, sir.
Okay, right, good. Um, so, one thing that we need to understand is every solution would have hydrogen ion if it's made out of water, but every solution will not have sodium ion. So, when do you have sodium ion in solution? Can anyone try answering this? When would any solution have sodium ion? Yes, Shreya.
Sir, in alkali conditions,
Okay. When you say alkali conditions, let's say you have a buffer. Let's say you have tris buffer. Now you want to know the pH of this tris buffer without before setting it, or let's say we talk about bistress propane. Before you set the pH, it's let's say 5. Now what would you add to it to make it to alkaline conditions?
sir, I think NaOH and something.
Right. You either add NaOH or KOH. So that depends on the salt that you have in your buffer. Yeah, this is how you introduce sodium in the first case. So NaOH shifts your pH from whatever it is to more basic, which is beyond 8. That is when you have sodium ions. And because your pH is measured in log terms, negative of log terms, the lesser you have an exponential decrease in hydrogen ions as your pH increases beyond 8 or so. That is when you first in the first place you introduce sodium ions. And once you introduce sodium ions, like you had mentioned, the glass electrode, it would mistake sodium for hydrogen. And this could al- you can try to associate this based on the valence electron that you have between sodium and hydrogen. 281 is how the electronic configuration for sodium is. So, and for hydrogen, it's just 1. So, it could very well be because of this one valence electron. So, now your electrode mistakes sodium for hydrogen. So there are two reasons. One is concentration, and the other is the valency of the element itself. Now, because it is mistaking sodium ions for hydrogen, you already have lesser hydrogens. Now, how would it affect the pH? Can anyone answer this?
Can you repeat the question, sir?
Yeah, so what I was asking is, in a given solution, you put your electrode is mistaking sodium ions for hydrogen ions. So, compared to your actual pH, is your pH lowered or increased? When you have sodium error, how is the pH changed? Is it increased or decreased? That's a simple way to ask this question. Okay, Ruhi says pH will fall. Ananya says pH will be high. Just think about this. You have this electrode. It is not. You have more of alkaline ions. You have sodium ions, and that is what is getting detected as hydrogen. So it is detecting more hydrogen than what is present. So if you have more hydrogens than, so let's say your electrode is thinking you have more hydrogens than what is present, will your pH be of lesser value? Lesser value as in, will it be towards 7 or will it be towards 10? That's what I'm asking.
Lesser value.
Right, yeah. So it would mistake and it would think that, yeah, high pH. Sorry, what is, yeah, Ruhi says it's 7. Yeah, Ananya, can you unmute and tell why you think it will be high? And followed by that, even Sri Lakshmi can try. Yeah, Ananya or Sri Lakshmi, do you want to mention why you think it will be high? Okay, seems like they are not here. Fine, but I hope you understood it. It mistakes and gives you a lesser pH, thinking there are more hydrogen ions, but there is actually more of sodium ions. Right. The next question is, cleaning optically polished glasswares with ethanolic KOH or other strong bases will cause which of the following? Decontamination, etching, sterilization, or sanitization. You can think about this question for a couple of minutes and then answer this. Yeah, so the question is, cleaning optically polished glasswares with ethanolic KOH or strong bases will cause dash decontamination, etching, sterilization, or sanitization. So, simply put, this optically polished glassware could be any of the cuvettes that you use with spectrophotometers, CD, or UV spec, any of these applications. Yes, it is etching, right? So here, so what is etching? Do you know? Do you chemically what is happening when etching is occurring? Ruhi Nishat?
Sir, excuse me, sir.
Yeah, you can talk. Yeah.
Sir, there is one etching process that I know. It's like shaping the glasses.
Okay.
But I don't know it in the context of chemistry. I know it in the context of making tools. I heard it in the television once. Maybe it's the same thing.
Okay. So
They etch the glass to make tools, sir, to make sharp tools. Maybe that's. I'm a bit confused about
no, no,
whether it's the same etching.
it's a common terminology that is that people use. You are
Okay, sir.
right. It's
Okay.
in this context again. Etching means you're basically damaging the surface by
Okay.
degrading the surface. People use it for in different contexts. For example, in nanotechnology, when people want to make these small microchips that
Right.
they add chemicals in minute quantities or apply lasers to give heat to specific positions in such a way they are able to carve it. So here what we are discussing is like what Shreya and Ruhi are mentioning, it's degradation. And so your glass is made of silica, and that when it reacts with this ethanolic KOH, it causes corrosion, which in technical terms is what we are calling as etching. And it is mentioned that it can damage your clarity and precision of your glassware. Right. The next question is glass pipettes with protein contamination can be washed with option A nitric acid, option B sulfuric acid, option C carbonic acid, option D chromic acid. Yeah, Ruhi, you can unmute.
Hello, sir.
Yeah, you can mention. Yeah.
So, answer will be chromic acid, sir, because that's a strong oxidizing agent than nitric
What
acid,
is chromic
sulfuric
acid?
acid.
What is chromic acid? How can one make chromic acid?
Um, how to make a chromic acid? But
Because chromic acid is not usually present. Rather, you would add a
use
couple of
in
things.
labs, laboratories, they use them for cleaning these things.
Okay. Why specifically chromic acid? Why not sulfuric acid or nitric acid?
So specifically, if protein contaminations are generally small residue of protein, if it is present, that may cause false report in the in lab data or something.
No, no.
So if we are using,
What I'm asking is, you
yes,
have
sir.
other acids as well: nitric acid, sulfuric acid, carbonic acid. Why do you
Yes, sir.
specifically want chromic acid? Shreya, we'll wait for a couple of minutes, or Or do you want to think, Ruhi, and then answer?
Yes, sir. I'll think about it. So actually, I thought that the strongest one, so we can use it.
What about sulfuric acid? Is it not strong enough?
I'll think about it.
Among these options isn't sulfuric acid the strongest acid? Okay, you can think about it. No issues. No
I'll think
issues.
about it, sir.
No
Okay.
issues. Yeah. Yeah, Shreya.
So I would like to add that sulfuric acid is highly corrosive because it's very strong. So we
Yeah.
shouldn't use it for cleaning glass pipettes.
Okay. So that way you say we can avoid using sulfuric acid. Okay.
Yes, sir.
So you agree that sulfuric acid is the strongest acid here.
Yes, sir.
Okay. Okay. And Srihari, you have mentioned nitric acid. Why would you say so? Okay, if it's an equally strong acid, wouldn't that corrode your glass pipette? Okay, you're saying avoid glass etching. Okay. I can tell you what I know about proteins. So, okay, why would proteins actually bind to glass? You have specific amino acid residues that are hydrophobic. You have hydrophobic patches because you have some biomolecules like fatty acids which are hydrophobic, and the way these interact with proteins is through hydrophobic interactions. And so naturally you have these hydrophobic amino acids which interact with your glass surfaces. So how would you ensure that these hydrophobic amino acids don't anymore interact with your glass surface? So you can think about this in terms of the chemistry of the molecule. One way to change the properties of amino acids within a given protein is by changing the properties of their side chain. And specifically here, if we think, oxidizing is one phenomenon that can change the chemical nature of these amino acids. Although Ruhi, I think, had mentioned, yeah, she had mentioned that chromic acid is a strong oxidizing agent. That is the reason why chromic acid is used and not nitric acid or sulfuric acid is used. Because here, if you see, when you add a dichromate salt with sulfuric acid, yeah, when you add a dichromate salt with a sulfuric acid, it becomes an oxidizing agent. And this oxidizing agent can oxidize these amino acids, thereby ensuring their chemical properties change, which would ensure that they no longer stick to the glass. That is why it's specifically used to remove stubborn proteins. Stubborn proteins are proteins, in this context, stubborn proteins are those that have these hydrophobic patches, meaning they have multiple residues of that are hydrophobic in nature, and this is the reason they stick onto glass. I hope that was clear. Okay. The next question is, what are pyrogens? And four different options are given here: inflammable contaminants, waste products, metabolic byproducts of microorganisms, antipyretic products. For this question, again, whoever knows the answer can raise your hand. You can think for a couple of minutes, think deeply, and then it's a simple question. You might have read about this even in your school level. Yeah, Shreya, you can talk about one incorrect option.
So, sir, inflammable contaminants is wrong because pyrogens are basically those that causes fever. So it is somewhere related to microbes. So I think inflammable contaminants is wrong.
Okay, what are inflammable contaminants?
Uh, sir, inflammable the things those types of foreign substances that causes inflammation, but it is not necessary that they cause fever. It can be redness or some kind of things.
Swelling, histamine.
Hmm.
Right. Good. Okay. And that depends on person to person. It could be a side effect of your allergies. Right. Uh, Anyone else want to mention something that you know about pyrogens and why a particular option here could be an incorrect option? Okay, one thing that I would like to, yeah, like Meghana had mentioned, uh, antipyretics are those tablets that you take to reduce fever. So that is also wrong. And let's discuss about what these metabolic byproducts of microorganisms are. So, in a microorganism, even if you kill it, there could be some proteins which could induce fever. So, even if it is dead, or there could be something specifically within the microorganisms that you don't have an immunity against, which could cause fever in you. So, here the right option would be metabolic byproducts of microorganisms. Anything to do with microorganisms. So, like it was mentioned earlier, pyrogens are these fever inducing substances. So, typical examples would be this LPS from cell wall and the metabolic byproducts that are components that are released when bacteria die. So, because the one thing to remember is that because these are just chemical moieties and that can induce a reaction in your body, even if they are dead, the nature of the pyrogen would induce a reaction in your body leading to fever. So, in other terms, you can think of this as the oldest method of vaccination, where live or dead or attenuated microorganisms were given to attain immunity. Right. Next, we have a few calculation based questions. We have the solution. It's better if you practice it to make yourself familiar with it. So the first question is, to prepare 80 ml 2% agarose gel solution, the amount of agarose required is dash. Four options are given here: 1.6 gram, 2 mg, 1 gram, 1 mg. Yeah, you can maybe take out a pen and paper and then calculate. Okay, the first thing to calculate, to make it easier, the first thing to calculate is calculate how many grams of agarose you need for making 100 ml of 2% agarose solution. And whoever has done that can answer this question first. how much agarose do you need for 2% of 100 ml agarose? So the question that I'm asking is for 2% of 100 ml agarose. Yeah, Shreya.
So for 2% in 100 ml, we need 2 grams of agarose.
Right. Yeah. So I have the calculation here. It's very simple. You might have done this in your school level. Yeah. So for 2% in 100 ml, you need 2 grams. So you just cross multiply how much you need for 80 ml, and the image is not very clear now. And if you need it for 80 ml, you just cross multiply this and you get 160 by 100, which is 1.6 grams. Yeah, Akshita, Rana, and Meghana. Yeah, both of you have given us 1.6 gram. That's the right answer. Good, all three of you. The next question is, to prepare a 100 ml NaCl solution of concentration 10 millimolar, the amount of NaCl required is dash. Molecular weight of NaCl is given as 58.44. Given the answer here, but it's better if you calculate it. I'll show you how to calculate it later. Does anyone know what molarity is? Molarity, anyone? What is molarity? Okay, molarity is the number of moles that you have in a defined volume of solution. So here they say the concentration is 10 millimolar and you have 100 ml of solution. So how much of NaCl is needed is the question. So how you calculate this is molarity is number of moles by volume, and the number of moles is the amount of substance that you have divided by its molecular weight. The amount of substance is given as GW here, given weight. That's how I write this. So you have 10 millimolar equals given weight by molecular weight, which is 58.5. And volume should be in liters here. So 100 by 1000. So when you take this here, you get 58.5 mg. Okay, good. I couldn't see this. Sorry. Piyush Rai has given mole of solute in 1 liter solution. And right. Jaya Sai Ramya. That's not just the unit of concentration, but what is better is how much you have, how many moles you have in a liter of solution. That would be better. Yeah, Ruhi Nishat and Tara Panwar, both of you have given the right answer. Right. So this is how you calculate the amount needed to make 100 ml of 10 millimolar NaCl solution. Next question is for a reaction A plus B equals C. Now the equal can be considered as a reversible reaction here. What is the K equilibrium? And 4 different options are given here. So can anyone tell me what K equilibrium is before we go to uh, the right answer for this question? Can anyone tell me what K equilibrium is? You can unmute and tell me.
The answer is D, sir.
No, you can first tell me what K equilibrium is.
Sorry, sir.
You can tell me what K equilibrium is. Yeah, so what is it? Okay, so who's this? Piyush Rai and Akshita Rana. Rather than just defining it physically, what does an equilibrium constant tell us? You have a reaction, like how it is given here. A plus B gives C. We also know that C gives A plus B. So what does this equilibrium constant KEQ tell us?
The ratio of products to reactants.
What does that mean? If you say it's the ratio of products to reactants, huh. So when you have a reaction that happens in both the forward direction and the reverse direction, you want to know at which direction. Okay, you first want to know if it is happening at the equal rate. If it's happening at the equal rate, X amount of product is being produced and X amount of product is being consumed at the same time. You want to know if that's happening, or more amount of product is produced, lesser amount of your reactants are produced, or vice versa. You want to understand this. So K equilibrium measures how fast is the forward reaction and how fast is the reverse reaction. It just tells you the rate at which the forward reaction happens compared to the rate at which the reverse reaction happens. Which is why you have K forward and K reverse, and this is how it is being represented. I'll come to the next question. That's what is given below. The question after this. Okay, the next question is the pH of the system is defined as. Okay, you would be familiar with this from your school level. It's the negative logarithm of H plus ions. Option A. And what does bicarbonate buffer help to maintain?
pH of blood.
Yeah, right. Any buffer that we use is helpful to maintain the pH range independent of whatever product is being formed. So it maintains the range of a pH at a particular range and doesn't allow it to be altered substantially. So here it means pH of blood. Right. The next question is trypsin has an optimal pH of dash 1.5, 3.7, 7.4, and 9.2. Okay, before we go to this, whoever wants to answer can start with mentioning what trypsin is and why is pH an essential thing and how would pH affect. So whoever wants to answer this particular question, you can start by answering what trypsin is, where is it found in the body, whatever you know about trypsin. Yeah, Shreya.
So I think trypsin is a protease enzyme that is basically found in somewhere like pancreas.
Is it found in pancreas or is it synthesized or produced in pancreas?
I think it's synthesized in pancreas.
Okay, it is synthesized in pancreas. Good. Okay. What do you
And we
remember?
also use trypsin in animal tissue culture, no, animal cell culture.
Okay. Where do you use it exactly?
Uh, for that adherent cells, when we want to like remove them from the surface, so we give trypsin, we do trypsinization.
Right. So you when you so okay. let's just make it clear for everyone. So in cell culture, you have two modes of growing. You either grow a suspension culture, which means that your cells can survive when it is growing in a liquid medium and in rotation as well. But when you have adherent culture, here your cells have to stick on to the cell surface, a surface that is at the bottommost layer. So here cells grow as layers, and it can survive only if it is connected and grown as layer. So this is the adherent cell system. And when you use trypsin, when you trypsinize your adherent cell culture, it is basically detached, and you can take your cells away. That's what who Shreya mentioned. Yeah. Right. Okay. Does anyone else know anything more about trypsin? Okay. So trypsin, I'll tell you what I know about trypsin. Trypsin is produced by pancreas, and it starts as an inactive trypsinogen. Then it gets cleaved in the small intestine by enterokinase. So it functions in duodenum. And okay, before I mention the other things, so it functions in duodenum, which means that duodenum has an optimal pH. Now, coming to the question, why would an enzyme require or function optimally only at a particular pH? Can anyone try to answer this question?
So enzymes so enzymes have active sites, then only they are functional. So at a particular pH, only these enzyme sites are functional. Because if we increase or decrease the pH, that may cause ionization of some of the amino acid that will disturb the active site, rendering the enzyme to be inactive. So at the optimal pH is needed for the proper functioning of enzyme.
Okay, and does it also have something to do with the structure of the enzyme?
Yes, sir, the structure will distort if we increase or decrease. Suppose if that causes the ionization or it disrupts the bonding between the amino acid or some of the amine group or carboxylic group in the amino acid may get charged and it cause the disruption of the structure, sir. Denaturation may be because
Right,
because
right.
of this.
Correct. That's the answer that I was looking for. So, enzymes are basically proteins, and proteins behave differently at different pH. And this is because one of the okay, we'll talk specifically about enzymes which have catalytic properties of other than being just a protein. So enzymes have these active sites which where a particular reaction happens. Now trypsin is a peptidase, meaning it breaks down peptides, or protease breaks down proteins, however you call it. Um, this process of breaking or cleaving of this peptide bond also needs specific coordination, and the specific coordination is not achieved at all pH and all temperature. It happens for a range of pH and a range of temperature, which is what I give as a graph here. If you see, pepsin is active at maximally at a pH 3, and then its activity reduces as it goes to pH 6, whereas trypsin, which functions in duodenum, it functions maximally at pH 7 to 8, which is around the pH range of duodenum, and it functions maximally at that pH. So the right answer is option C, 7.4. Finally, the last question is, a solution contains 0.35 molar sodium acetate and 0.45 molar acetic acid. The pH of the solution is dash. We have been given the pKa of uh, acetic acid, which is 4.74. So you might have read about this in your high school chemistry or even before that, there is a particular equation that people use to relate pKa, pH, and the amount of free acid that you have in a given solution. Does anyone remember what this particular thing is? Particular equation is called as? Yeah, Hasselbalch equation. Yeah, so it's called Henderson-Hasselbalch equation. Yeah, Piyush, what do you want to tell us what you know about Henderson-Hasselbalch equation? Piyush, are you there? Piyush? Okay, you don't have a microphone. Fine, I'll go on to the equation. So you have pH. So they say pH equals pKa plus log of concentration of this anion that otherwise forms your acid here. So in the given question, they say the concentration of sodium acetate and the concentration of acetic acid. So this is your respective anion and your acid here. So and pKa of this is given here. So 4.74 plus log of 0.35 by 0.45. When you do this, you get 0. Sorry, so you get 4.631. And so the right answer here is option B, 4.63. So with this, we come to an end of today's problem solving questions. If you have any doubt, you're free to ask me. This PPT will also be uploaded and the link will be shared with you all. You can check it later as well. If you have no doubts, you can So textbook, I wouldn't really have recommendations unless your course instructor has recommended. I can check once, but what I do know is give me a minute. Mentor course recommendations. You can check all these books. These are what is listed by your course instructor.
Sir,
Yeah. Yeah.
so earlier we discussed about etching.
Etching.
Right,
Yes.
sir?
Yes. Yes.
So, sir, etching, it happens because of like strong bases, right?
Right. Ethanolic KOH can cause etching. Yeah.
Right. So, sir, is it because of rubbing it or just keeping it will also cause itching?
No, no. Silica. This is a chemical reaction, right? You need not put physical pressure there. Although physical pressure can cause a different form of etching.
Yeah. So if we, where I'm going with this question is that we are well aware that we are supposed to, like we can keep acid in glass containers.
Okay.
That's okay, and that's not nothing in no problem with that. But what if we keep bases in glass containers? The
Okay.
etching
So
will
yeah.
also.
So the context here is you do not use the same glass containers for spectrophotometric readouts. When
Right.
you're doing spectrophotometric experiments,
Right.
you are trying to measure the number of photons that are being emitted. That should not be altered because of this etching. That is what
Okay.
people are worried about in this particular
Okay.
question that we discussed today. So as long as
Okay.
your etching doesn't affect your photon to travel and reach your sensor, you do not have to really worry about it.
Okay, sir.
Right. So I'll stay on the call. If you have any questions, you can ask me.